So today we're going to look at an important immunological component called the complement system. So basically, the complement system is made up of a couple of proteins that act together in a certain sequence to elicit an immune reaction. So these proteins normally are found in serum, and they do not increase after immunization or after exposure to a certain antigen or whatever the activation component is. So basically, it is an innate immunity. So the proteins are inactively circulating in the plasma. So even right now you have them, but they only become activated when they are cleaved into fragments. And then these fragments are the ones that make them now activated and they become functional. And the ultimate event is basically you end up having lysis of the cells, or in this case, the pathogen that we are looking at. So we have three main pathways. So the classical, the lectin pathway, and the alternative pathway. All of them normally come together at the point of formation of a C5 convertase, and thereafter you end up having a membrane attack pathway. So we have certain other elements or responses that we end up having. However, the main thing that we have for the complement system is membrane attack pathway, which leads to lysis of the pathogen. So in this case, we will try and focus at the classical pathway, and each one of them has specific elements that actually activate them. So like in this case, we'll talk about classical pathway, which requires, it's an dependent on antibody antigen binding. So the classical pathway basically is activated by an antigen antibody complex. So you have a bacteria, for example, or whatever pathogen that reacts with an antibody, and then they form an antigen antibody complex. And that is what initiates or activates the entire thing. Now, it is important to note that there are some antibodies that can work for the classical pathway, not all of them. So IgG, or immunoglobulin G, and immunoglobulin M, these are the only ones that have the fraction of crystallization, the FC portion, have the receptors for the proteins for complement. So the other ones do not have, therefore they will not initiate the classical pathway. So you require IgG or IgM for this pathway to proceed. So the activation of, you have talked about proteins. So normally we have C1 to C9 proteins, and they follow each other in that fashion. There's some differences that occur in between here, but typically we have C1 to C9 proteins. So typically what we have is that we have a bigger part, which is a bigger subunit, normally designated as B. So actually this B should be here. And then the smaller portion is A. So this should be A. The only difference is in the case of C2. So actually what I've drawn here is for C2. So for C2, the bigger portion is normally the A part, and the smaller portion is the B part. So I repeat again, typically the biggest subunit, typically for almost all the other proteins, the biggest subunit, this bigger one, is normally B, and the smaller one is normally A. But in the case of C2, C2 is the only exception, where we have the bigger portion being A and the smaller portion being B. So that being the case, the heavier portion, or the protein, is the one that is dropped on the cell surface of the pathogen, and the smaller portion is the one that is left to go in the blood back into the serum. So therefore, as you'll see going forward, it will be actually having a complex being formed by the bigger portions. So if we start off the pathway of the classical pathway, so we have C1. So C1 is the first part. So C1 is basically a complex of 3 subunits. We have C1Q, C1R, and C1S. So these ones are R, and then we have S here. So basically the point here is that C1 is made up of a complex C1QRS. Okay, so for these molecules, for these subunits to bind, they normally require presence of calcium to actually hold them together. As I've said earlier, this pathway requires antigen-antibody complex. So this is our antigen, or the cell surface of a pathogen. And then we have our immunoglobulin. So this is IgG, and you can see the Fc portion, which is binding, which is a receptor for the binding of C1. So the C1q activation requires binding to a C1q specific receptor on the Fc portion. So it must have, there must be an Fc portion. So as I said earlier, this can only happen in IgG or IgM because IgA and IgE, they do not have receptors for the complement proteins. So therefore, so once this binding happens, the C1q arrests. So the C1q activates C1r, and then in turn, C1r will activate C1s. So that is the sequence of events. So that's why it's called C1QRS. So Q activates R, R activates S, and then it is from there. Then we have S activate or acting or cleaving. So when we be talking about cleaving, it's just like cutting up or chopping up into two portions. Remember the bigger portion and the smaller portion. So the C1s, which is now the final activation of a C1 complex, it now cleaves C4, splitting it into two fragments, C4a and C4b. Again, if you remember, we said in all the other proteins, B is the bigger chunk and A is the smaller chunk, except C2. So in this case, we'll have C4a and C4b, and as you can guess, C4b will be the larger chunk or the larger moiety, which will basically be the one to continue. So as you can see here, we have the C4b, which is bigger, C4a, which is smaller, so C4b will drop on the surface of the cell. Then the C4b complex with, or basically it reacts with the C1s to also cleave C2, or it acts on C2, and then it splits C2 into C2a and C2b. Remember, we said C2 is the only exception where the C2a is the larger moiety in this case, or the larger chunk. Therefore, C2b is the small one, and the C2b goes back to serum or blood, and then C2a drop. So we have now therefore formed C4b, which is the bigger chunk for C4, and C2a, which is the bigger chunk for C2. Therefore, the C2a together with the C4b, they will merge together to form C4b2a. So we just remove the C. We don't keep on repeating C4b, C2a. We just say now C4b2a. So this C4b2a, remember, comes from the 4b and the C2a. Okay, so that forms a C4b2a, which is also called a C3 convertase. Okay. Now the C3 convertase works by cleaving. So a convertase is an enzyme. So the C3 convertase splits C3. Okay, so remember the C4b2a was the C3 convertase. So it splits C3 into C3a and C3b. Okay, remember again, C3a, C3b is the bigger chunk, therefore C3b will drop and it will go to the cell surface, and then it will merge together with what we had C4b2a to form C4b2a3b. So there's a B missing here. So this complex here that we have formed is called a C5 convertase. So if we use this to go back to what we said, so we started with the C1QRS, cleaved C4 into C4A and C4B. So we had C4B here, and then cleaved C2 into C2A and C2B. So C4B and C2B came together to form C4B2. Actually, this is supposed to be 2A, to form C4B2A, and then this actually leads now to formation of a convertase, also breaks down C3 into C3b and C3a, and then C3b is taken up. Okay, so just to clarify matters here, this is supposed to be C4b2a, okay, 2a, then 3b. So this C4B2A3B is what we're calling a C5 convertase. And as always, a convertase is an enzyme which will help split C5 into C5A and C5B. And as you can guess, C5B is the molecule that we'll continue with. So C5B binds to cells to activate. So it binds now to the cell to activate now the remainder of the proteins. So it activates C6 and C7. So the complex C5, so we'll end up having C5B. Remember, we had C5B, and then 6, 7. These activate C8 and C9, so that they all come together to form C5B, 6, 7, 8, 9, which is now called a membrane attack complex, the MAC. So this is basically C5B activating C6 and C7 here. And then we end up forming a C5B67 complex, which activates the binding of C8, then C9, and then we have this complex. As you can see, this complex is like a hole that has been formed here. And this hole is basically a hole that has been made through the cell surface. Okay, so this C5B6789, remember we called it a membrane attack complex, which is basically some sort of channels or holes, what we're calling a transmembrane channels that are formed on the cell surface, which allow ions to pass through. So this movement of ions creates a disequilibrium of the cell, disturbing the influx of water, so causing the cell to swell. Okay, as you can see, this was a live cell before the reaction, and then after the reaction, it has swollen, and then it starts getting lysed. As you can see, there's some swellings that are being formed here. So that is the classical pathway of the complement system. Thank you.